All right, so before we move on, let's first take a very quick look at the TCA cycle. Okay, again, this is TCA cycle, as you can see on the screen right here. TCA cycle, the input is 1-acetyl-CoA. Okay, that's the input, 1-acetyl-CoA. And acetyl-CoA, please just remember, acetyl-CoA is 2 carbons, 1 carbon being right here, 1 carbon being right here. Okay, so there's going to be 1 carboxylate carbon, 1 methyl carbon. Okay, so those are your 2 carbons right here. And those 2 carbons are going to react with oxaloacetate, okay, whereby you form a new bond between this carbon all the way with this particular carbon. Okay, so after you form this carbon bond, then you generate your citrate, okay, which TCA cycle is named after citrate because it has 3 carboxylates, which is called tricarboxylic acid cycle. Okay, so TCA. All right, so this is citric acid. Now, citric acid, if you look from the structure, okay, and a lot of people, like I said, will always choose to write it this way because it's easier to remember, regardless of how you write it. Okay, TCA cycle. First intermediate is citric acid. Okay, citrate. Now, citrate. If you look at citrate, you'll see that citrate is pretty symmetrical, which makes citrate not a chiral molecule, so it's achiral, right? It's achiral. And then what you do is you use an enzyme called aconitase. Okay, and by the way, here is citrate synthase. Okay, and using aconitase, you will always move the OH here to the pro-R carbon. Okay, on the other side. So pretty much this carbon. Okay, this one. And then essentially what you'll generate is isocitrate, which a lot of times people will draw it this way. Okay. All right. After you get isocitrate, okay, and then you do a very simple oxidation. Okay, so you oxidize this particular OH into a carbonyl. Okay, and because you do that, you now have, let's say here is the carbonyl carbon, then here is alpha, and that would be beta. Okay, so here, this guy in the middle becomes the beta carboxylate, okay? Becomes the beta carbon. And like I said, beta carbon, if you do a decarboxylation on the beta carbon, you will get a stable intermediate, okay? So because it has a resonance, so that beta keto acid in general is unstable, okay? So you will kick out this particular CO2 such that you get you get alpha ketoglutarate, okay? And this thing is called isocitrate dehydrogenase. Okay? So its primary function is doing an oxidation, dehydrogenation of isocitrate, but because in the process you will also create a beta keto acid, and beta keto acid in general is less stable, so the final product you get is a decarboxylated product. Okay, so you get alpha ketoglutarate. And remember that now alpha ketoglutarate, this guy, is now an alpha keto acid. Okay, and you want to turn an alpha keto acid, okay, this is 5 carbon, into a 4 carbon acyl-CoA. Okay, so this reaction is going to be identical to your pyruvate dehydrogenase complex. Okay, remember that? And remember last week we talked about how this enzyme also has 3 components, or the 3 functional enzymes inside of this big complex, and there's the E1, E2, and E3, right? And E1 and E2 are different from PDH because this guy is going to accept a 4-carbon intermediate inside it, whereas PDH is only transferring 2 carbon, right? Remember we talked about that? But E3, you can use the same polypeptide, okay, the same protein, because E3 does not involve your carbons, okay? It's only doing oxidation reduction to convert the 2 thiols back to the disulfide bonds, okay? So pay attention to that and review it a little bit. And then this guy obviously is going to be called alpha Kg, alpha Kg, okay? Alpha Kg dehydrogenase. Okay? And then you get down to succinyl-CoA. Succinyl-CoA, now this guy is a high energy thioester. Okay? And you go from high energy thioester to acid. Okay? So the delta G is going to be negative, right? So it's going to be around -30 if you do only a hydrolysis. So evolution has figured a way out to conserve that energy. So then you're going to use an enzyme, which is called the succinyl-CoA synthetase, okay? Which then essentially take the thioester energy and save it into ATP, okay? Or in the case of mitochondria, it's GTP, but pretty much the same thing. All right, and then you get to succinate. Now, succinate, this thing, again, it's going to be achiral, right? It does not have chiral center, and it's going to be symmetrical, okay? So carbon 2, let's call this guy carbon 2, carbon 3. 2 and 3, you cannot distinguish, right? And after that, you would do a oxidation to make fumarate, okay, by introducing a double bond right here. Okay, so remember, the more double bonds you have, the molecule is more oxidized. Okay? So at fumarate, C2, C3, also you can't differentiate. And you would do a hydration of fumarate to generate malate. All right, and here, malate, you can have essentially 2 positions where you can do hydration. You can do it at C2, or you can do it at C3. Doesn't really matter, okay, because fumarate is symmetrical, okay? It's symmetrical, so you can do the hydration at C2 or C3, right? The enzyme cannot tell, okay? The enzyme will not be able to know which one is really C2, which one is C3, okay? Regardless, then you'll generate malate, okay, where you will have this hydroxyl group right here, and then after that, you will then oxidize malate OH, okay, the malate hydroxyl group, into a keto group for oxaloacetate, okay, or in short, I usually just call it OAA, okay, but actually it's not like that. It's oxaloacetate or oxaloacetic acid, okay, so it's acid, okay, so OAA. All right, so this is the functional cycle of TCA cycle. Okay, now if we look at the energy, okay, you saw this last time. If we look at the energy, what you should see is that the standard energy, there are 3 that's very negative, okay, being steps 1, 3, 4, okay? Steps 1, 3, and 4. Step 1, it's very important because if you control step 1, then you control the entire TCA cycle, okay? If you don't have acetyl-CoA coming in, then you don't have a functional TCA cycle. Make sense? Okay. So that the energy here is also -31, so you would control this one as well. What you should see is that steps 3 and 4, you have isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. For those 2 steps, the delta G standard delta G is also quite negative. Okay, and you know, it should also make sense because in those 2 steps, you have decarboxylation. Okay, you kick out CO2. All right, so any steps that you kick out CO2 is in general very favorable reaction. Okay, so you'll also want to control these 2 as well. Okay, And the only one that's kind of weird is malate dehydrogenase. The standard energy is positive 30, but we can sort of tune the concentration of OAA, concentration of malate, such that inside the cell we can get a delta G of close to 0, okay, so that this can actually proceed. All right. So again, in terms of regulation, you should be very comfortable understanding that TCA cycle is a cycle used to generate energy. Okay. It's also a cycle used to generate intermediates for producing some amino acids. Okay. Therefore, okay, if you have a lot of ATP, you don't want to run TCA cycle. Makes sense. Okay. It makes sense. If you have a lot of TCA cycle intermediates, you don't want to run TCA cycle. Okay. So it's very intuitive, such a way that if you have too much of the product, you're going to shut the cycle down. So for example, citrate synthase, this guy, is going to be inhibited by citrate, okay, and it can also be inhibited by succinyl-CoA. Okay, so products of the TCA cycle can then come in and inhibit your TCA cycle. All right, so we talked about that. All right, and last time we pretty much stopped at Anaplerotic reactions. Right, so before we move to there, just a quick reminder that you have two types of reactions. One is the cataplerotic and one is the anaplerotic. Okay, cataplerotic are the reactions that will consume the TCA cycle intermediates. Anaplerotic reactions are the ones that will sort of regenerate the TCA cycle intermediates. Okay? All right, so the four major anaplerotic reactions, these I need you to memorize these. Okay, these are very important because of the fundamental reason that these are carboxylases. Okay, they can fix CO2. Right? You have PEP carboxylase. This is used a lot of times. Okay, so it's used by many different organisms. And essentially what it does is it uses the energy from PEP. You can directly capture a CO2, bicarbonate CO2, same thing. Okay? And then you will put that CO2 at the very end, and then you generate oxaloacetate. Okay? Or additionally, you can sort of save a little bit of energy and you produce ATP at the end. Okay? This is PEP carboxykinase. Okay? All right. It has a kinase at the end because it's using ATP. Okay, so it's why it's called PEP carboxykinase. Now, this guy we saw previously in gluconeogenesis. Okay, but going in the reverse direction. And then you also have pyruvate carboxylase, which uses ATP, and you use ATP as the energy source to then be able to put the carboxylate, the CO2 right here, onto pyruvate right here, right? And then lastly, you have this thing called the malic enzyme, okay, which is essentially using the formation of malate to drive this carboxylation, okay, of pyruvate to malate, okay? Now, this last reaction actually makes sense because if you look back from TCA cycle, okay, if you look at the energy of TCA cycle, remember we said that malate to OAA is like plus 30, okay? So the reverse is going to be OAA to malate, that's going to be minus 30, okay? So pyruvate carboxylation, okay, so if we put pyruvate with CO2 combined, you will generate oxaloacetate, right? Make sense? Okay? And in one step, if you take oxaloacetate to malate, you have a minus 30 of energy. Do you guys know what I'm talking about? Maybe? Maybe okay? Okay? So essentially, pyruvate, you can think of it as pyruvate become OAA, OAA become malate, okay? And remember that this step, delta G, is going to be approximately minus 30 kilojoule per mole, right? So essentially, you'll use that to drive your reaction, okay, because the formation of malate is more stable than the formation of oxaloacetate. Okay? So you can think of it that way. All right? And we talked a little bit about amino acids, but we'll come back to the synthesis of amino acids and TCA cycle intermediates. But essentially what happens is that alpha-ketoglutarate and oxaloacetate, they participate in a lot of amino acid biosynthesis and degradation. So we'll talk about this when we talk about amino acids. Okay? And here, what I just want to show you is that pyruvate pool turns out to be very important for TCA cycle, okay, because it can regulate TCA cycle, right? Because if you think back to our TCA cycle and our Anaplerotic reactions, both acetyl-CoA and oxaloacetate can essentially be provided by pyruvate, okay? So let's say today you turn a bunch of pyruvate into acetyl-CoA, okay? So you make a lot of acetyl-CoA, but you don't have enough OAA, okay, not enough oxaloacetate. Then TCA cycle is not going to be running at a very fast speed, okay? So as a result, you need to then use pyruvate to put more flux to oxaloacetate, okay? So turns out acetyl-CoA, okay, acetyl-CoA can inhibit the enzyme that synthesizes itself, okay? And it can activate pyruvate carboxylase, okay? Does that make sense? If you have pyruvate going to acetyl-CoA too much, then you have a bunch of acetyl-CoA, not enough oxaloacetate, not enough OAA, and then acetyl-CoA can then in turn inhibit PDH, activate pyruvate carboxylase, okay, which will synthesize you OAA. Make sense? Okay, so it's sort of like a little control thing that it can do. Okay. So at the very end, last lecture, we talked about carbon labeling, okay, carbon labeling. So when you form a new bond between acetyl-CoA and oxaloacetate, you essentially transfer, so this guy, remember, it's the same thing as acetate, okay? Remember that acetyl-CoA is nothing more than just an activated form of acetate. So then when you put acetate onto oxaloacetate, okay, acetate onto OAA, you will put it essentially right here, okay? And remember that isocitrate, okay, the OH is always going to go to the here and not here, okay? So the OH migration will always move to the carbon that's, you know, opposite of the acetyl-CoA, okay? And then subsequently, your labeled carbon is going to be right here. It's going to be right here. Now, note that because succinate and fumarate, they're symmetrical, so C2 and C3 cannot be distinguished, okay? So right here, essentially you have this one labeled as well, this one labeled as well, okay? And when you go down to malate, now you have two kinds of malate, okay? You have one malate that's labeled right here, another kind of malate that's labeled right here, okay? So if you analyze it using a mass spectrum or mass spectroscopy, essentially what you'll get is you will get two different peaks, okay? You will have two different chemicals. One is a labeled malate that's on C2, one is a labeled malate that has the label on C3, okay? And then same with oxaloacetate, right? So labeling will make a difference. Now, so here I actually want to show you a couple of things. I want to show you the fate, the actual fate of acetyl-CoA once it's entered TCA cycle, okay? Where do the carbons of acetyl-CoA end up? Obviously CO2, but in one round or in two rounds, okay? So pay a little attention here, okay? So essentially the question that you have to ask yourself is how many times the TCA cycle has to operate to oxidize a particular labeled acetyl-CoA to CO2, okay? All right, now let's first look at first round, all right? TCA cycle. your acetyl-CoA, let's say we label this guy and this guy, okay? We label this acetyl-CoA. Okay, we can feed a C14 labeled acetate or C13 labeled acetate to the cell, and the cell is going to start incorporating these acetate into the TCA cycle, okay? So let's see where these things end up, right? So the first reaction, we'll make it right here, okay? Make it right here. After you do the aconitase, okay, it's still right there because OH essentially goes from the middle to the bottom, okay? Your acetates are still right here. And you do decarboxylation, doesn't affect your acetyl-CoA, okay? Do you see that your the 2 carbons from your acetyl-CoA is untouched, okay? So you do another decarboxylation, your acetyl-CoA is still there, okay? The 2 carbons that are labeled is still there, okay? And you go from suc-CoA to succinate, succinate to fumarate, okay? Now, what you should notice is that from succinate to fumarate, now the carbon initially on top, okay, is indistinguishable from the 2 carbon on the bottom because succinate is symmetrical, okay? Because it's symmetrical, so you cannot tell the difference between the 2 carbons on top or the 2 carbons on the bottom. You can't tell. Therefore, by the time you get to malate, you also can tell. Okay, you also can tell. By the time you get to oxaloacetate, you also can tell. Okay, so now by the time you go to oxaloacetate, your labeled carbon, okay, the 2 carbons, okay, your 2 labeled carbons could be anywhere. Okay, it could be either C1, C2, or C3, C4. Okay? Alright? So like I said, unlike citrate, succinate does not differentiate between the 2 carboxylates for subsequent reactions to fumarate and malate. So labeling here is the same thing as saying labeling here. Okay, now. So at the end of one round of TCA cycle, the labeled carbon of acetyl-CoA end up at either C1 and C2 or C3 and C4 of oxaloacetate. Okay? So here, my point is that, in the first round of TCA cycle, the 2 acetyl-CoA that you incorporated is not lost as CO2. Okay? The net reaction is 1 acetyl-CoA coming in, 2 CO2 come out. Okay, that's the net reaction. But the physical acetyl-CoA right there on the very top that you consume in is not lost as CO2 in the first round. Okay? Obviously, in the subsequent round, it will eventually be lost as CO2, but in the first TCA cycle, okay, the first time you do this cycle, those 2 carbons are not lost as CO2. OK， 懂我意思吗？这个 OK， 这很重要哦。就是我们 TCA cycle 有没有？虽然说我不是不断在跟你们讲说 TCA cycle 一个 acetyl-CoA 进来，出去2个 CO2， 对不对？这是在讲的是 net reaction。OK， 每一个 acetyl-CoA 进来，你就会有2个 CO2 出去，没有错。OK， 这是 net reaction。但是那2个 acetyl-CoA， 假如说我们特定的在看一个特定的 acetyl-CoA，OK， 那2个碳进来之后，你跑一次 TCA cycle， 你会发现到说你那2个碳并没有出去 as CO2， 懂我意思吗？所以你看那个 acetyl-CoA 不是有变蓝色吗？有没有？所以蓝色的东西进来到跟 OAA 做 binding 之后，你那个蓝色的那个2个碳会在最上面嘛，然后你不断的那2个碳都不会不见，有没有？所以就是 acetyl-CoA， 我一个 acetyl-CoA 进到 TCA cycle， 跑一次 TCA cycle， 那2个碳还在里面，就还在 TCA cycle 里面，它并不会被踢出去，踢出去的是上一个 round 的 acetyl-CoA， 所以这2个碳呢，接下来是下一个 round 才有机会被踢掉。OK， 所以但是不要 confuse 哦，因为我去年教这个课的时候，就大概有1/3的人 confuse， 变成说，因为我考试一定会问一个东西是，比如说一个 acetyl-CoA 可以给你几个 NADH， 然后有些人就开始想说，老师好像说 acetyl-CoA 要跑3次，它才会被变成 CO2 嘛，然后它最后所有东西都给我乘3，那就不 OK， 你懂我意思吗？所以你要 differentiate 我说 net reaction 跟真正发生在那个 acetyl-CoA 这两回事。net reaction 是一个 acetyl-CoA 进来，它是2个碳没有错，然后你整个 cycle 跑完，你也是真的是踢掉2个碳没有错 ，OK？net reaction， 但是你踢掉的那2个碳呢，是上一个 round 的 TCA cycle 的 acetyl-CoA。好 ，OK 哈，可以哈，好哦，不要搞混哦。OK。So if we look at second round, and let's say, in the case of labeled as C1 and C2, okay, if we look at, our label was at C1 and C2, right, because we said that at the end of the first round, the label can be at C1, C2, or C3, C4, okay? It cannot be C1 and the C4, all right? I hope this is clear. All right, so let's say, the 2 labeled carbon is at C1 and C2, okay? What will happen? So if you look at this guy, right, this guy right here, ends up right here, okay, after adding acetyl-CoA. And by a conatase, it ends up still in the middle, okay? Then, okay, remember that isocitrate dehydrogenase will kick out the carboxylate in the middle. So you lost 1 CO2 here, right? So then the label becomes 1 carbon in the middle. Okay? So initially you have 2 labeled carbon, 1 this guy, 1 this guy. Okay? And now in the second round of TCA cycle, you lost 1 as CO2. Okay? And then this guy, luckily, it's going to remain. Okay, so I'm going to erase this. It's going to remain right there. Okay, so the second round, you lose the initial acetyl-CoA, 1 of the carbons. Okay, ma? Okay? All right, so 1 of the labeled carbon from the first round, here I should say from first round, okay, is kicked out in the second round. Okay, and by the third round, it's here, it's here, it's here, it's here, still there. Okay, so third round is still there. Okay, now it went from C3 to C4. Okay, all right, and then the fourth round, then you can then finally kick it out as CO2. Okay? All right, so my point is that the acetyl-CoA, the 1 acetyl-CoA that goes into your TCA cycle, okay, does not get kicked out as a CO2 until maybe second or third or fourth round of TCA cycle. Okay? 懂我意思吗？所以不是说2个 acetyl-CoA 这样 boom 这样飞进来，然后你那2个 acetyl-CoA 直接变成 CO2， 不是哦。OK， 所以你的那个 acetyl-CoA 它其实是会被convert 进去你 TCA cycle 的 intermediate 的碳。OK， 只是说我们在讲 net reaction， 就是1个 acetyl-CoA 进来，2个 CO2 要出来。OK， 只是说那2个 CO2 出来的不见得是刚刚好进去。OK，make sense？ 好。所以那个回去啊，想一下这个 ，OK。所以我这边会有一个小东西我漏掉没讲，就是我刚刚不是说 after first round，OK，after first round 之后 ，your OAA，your labeled acetyl-CoA, it's either on C1 and 2 or C3 and 4, right? For label. And here I showed you what happened if we say the label is at C1 and C2. OK， 你回去想一下。如果你的 label 在 C3 跟 C4 的时候，那它在第几个 round 可以把所有的碳排出来？所有原本进去的 acetyl-CoA 提出来，懂我意思吗？懂我意思吗？就是我这边给你的 example 有没有？我们不是说第一个 round 进来的时候 acetyl-CoA 进来，然后第一个 round 有没有？然后你最后你的 final product 是 oxaloacetate 嘛，对不对 ？OAA。然后你的 OAA 呢有4个碳，那你的 label 呢可能是1、2或者是3、4，对不对？然后我刚刚这边的 example 是说， 如果我们看你的 label 在1、2，然后会跑几次，然后你才能把这个1、2的 label 的碳排出去，对不对？那我没有给你看3、4哦，对不对？所以你回家想一下，3、4会长什么样子？在什么时候会被提出来？其实 from 这个 example it should already be pretty straightforward 啊。OK， 我不知道你有没有发现一件事情，就是那个刚刚第3个 round 还是什么？第3个 round 就是你会在 C3 嘛，然后第4个 round 它跑到 C4， 对吧？所以它 position 是一直往下跑。不知道什么意思的话，回去看一下，好吧？可以吗？ Alright, so now, this is actually the last thing that I'm going to talk about for today. It's actually quite important. Okay, this is very important. This thing is called the glyoxylate shunt. Okay, glyoxylate shunt. What it is, it's a bypass. Okay, it's actually a bypass in TCA cycle to avoid CO2 loss. Okay, what does that mean? Okay, what does that mean? Now, a typical TCA cycle, you have citrate to isocitrate, isocitrate eventually to succinate, right? This is the TCA cycle. And in the TCA cycle, here you will lose 2 CO2, right? You go from C6 to C4, okay? So you lose 2 carbons. Now, let's say I don't want to do that, okay? I don't want that, okay? So turns out there are some organisms on Earth, okay, particularly bacteria, okay, and fungi. What they can do is, once they get to isocitrate, they can do an enzyme, okay, they can use an enzyme called the isocitrate lyase, okay, so we'll chop isocitrate in half into succinate, and this thing called glyoxylate. Okay? Alright. Now, succinate can then continue to go to oxaloacetate. Okay? So essentially, what this pathway did, was instead of 2 CO2 for the normal TCA cycle, you make that 2 carbon into glyoxylate. 懂我意思吗? Okay, so instead of kicking out 2 carbons as CO2, what the glyoxylate shunt does is it's going to convert isocitrate to succinate, but releasing glyoxylate. Okay, so this is 2 carbons, right? 1, 2. Okay, 2 carbons. 1, 2, and then CO2 is also 2 carbons. Okay, so instead of emitting 2 CO2, I will save the carbons and of course the energy inside of the carbons, okay? Save the energy, and the energy is going to be stored in glyoxylate, okay? Glyoxylate. Okay, now glyoxylate, alright, turns out can react with another acetyl-CoA, 2 carbons, okay, so 2 carbon plus 2 carbon, guess what? It can give you malate. Okay? 来咯，很厉害哦，这真的很厉害哦，真的很厉害哦。Okay. All right. Malate can then become oxaloacetate, right? Okay. So what happened? Okay. What did you do? What's the net reaction? So if I give you this pathway and I want you to tell me the net reaction, what do you think the net reaction is for the glyoxylate shunt to generate essentially your, okay? Like net reaction. What do I consume? 所以我们先说 net reaction 嘛，就是说我吃什么，然后吐什么嘛，然后你再去 cancel， 然后最后就是你可以得到你的 net reaction. Okay, so in this pathway, we don't want to talk about TCA. Okay, so we only talk about the glyoxylate shunt. All right, so in the pathway here, what's the net reaction? What do you consume? Crystal, what do we consume? Acetyl-CoA. How many? 2. Okay, we consume 2 acetyl-CoA. Okay, and we also consume 1 oxaloacetate, right? Right. So 1 oxaloacetate plus 2 acetyl-CoA. Okay? What's your product? 2 oxaloacetates. Okay? So obviously the oxaloacetate can cancel out, right? So the net reaction is 2 acetyl-CoA to 1 oxaloacetate. Okay? Right? So here, let me write this down. So here you have the reactant. You have a, what is it? OAA plus acetyl-CoA plus acetyl-CoA. AC-CoA, I just mean acetyl-CoA, okay? Product 2 OAA. And of course you have some reducing powers. You have 1 FADH2, okay, right? Because succinate to fumarate. And you have 2 malate, okay, so you have 2 NADH. Okay, so this is the net reaction, and obviously you can cancel this guy with 2. Okay, so you have 2 acetyl-CoA giving you 1 oxaloacetate. So you convert 2 C2 to 1 C4. 可以吗? Okay. Now, remember oxaloacetate can then go gluconeogenesis to glucose. Okay? All right? Acetyl-CoA is the degradation product of fatty acids, acetate. Okay? It's the degradation product of acetate and fatty acids. So that if you have the glyoxylate shunt, you can convert fats to sugars. 我再说一次哦 ，OK， 如果你今天有 glyoxylate shunt， 你就可以把脂肪类型的东西跟醋酸 ，OK，convert 成糖 ，convert 成 glucose。如果你没有 glyoxylate shunt， 你就做不到 ，OK？ 为什么？你没有 glyoxylate shunt 的话，你的那2个碳就会在 CO2 了，所以你再怎么样你也做不到2个 OAA， 那一个会 recycle 回去 TCA cycle， 那一个可以跑掉 ，OK？ 所以如果你今天没有 glyoxylate shunt， 你是没有办法把这个 acetyl-CoA convert 回去 pyruvate。OK， 当然有一些很神奇的 bacteria 是可以用那个。有点像是 pyruvate 直接acetyl-CoA 直接往回跑到 pyruvate， 但是那个我们生化2不会讲到。Alright, so this is my final message, okay, most important message. Right? So for TCA cycle, okay, for TCA cycle, it's impossible to convert acetyl-CoA back to pyruvate using TCA cycle. 这很重要哦。因为你知道为什么很重要？因为去年我上这门课的时候，就一堆人在那边什么 TCA cycle 跑2次，然后就回去了。No. 不行啦 ，OK， 你跑2次不行啦，你那个碳会不见 ，OK， 会变成 CO2， 不可能啦，你跑几次都不可能 ，OK。所以如果你今天用的是 TCA cycle，OK， 就是 TCA cycle 哦，你 acetyl-CoA 进来一定是出去 as CO2，OK。所以如果你没有 glyoxylate shunt 的话，你是没有办法把 acetyl-CoA 变成 pyruvate，OK。那如果你今天有 glyoxylate shunt，你可以把2个acetyl-CoA, okay, 2 acetyl-CoA becomes 1 pyruvate. Okay, you lose a carbon in oxaloacetate to pyruvate. Okay, we saw that in gluconeogenesis. Okay, 可以吗？可以哈。Alright, so no. Here's the question: Do humans have glyoxylate shunt? No. Okay, well, at least not we know of. 就是现在。没看到 right? So primarily, so far, it's primarily found in bacteria, plants, and fungi. Or fungi, as some people like to say it. Fungi or fungi. Okay? So humans or animals, mammals. So far, some people have seen observations that suggests, but as of right now in the textbook, people don't say that humans have this. Therefore, remember like a while back I said that you cannot grow by drinking beer, for example. Actually, 扣掉，应该 beer 里面可能会有一些糖类。但是如果你今天只喝酒，只吃酒精，或者只吃醋酸，或者只吃脂肪 ，OK， 只吃 fatty acid， 你是没有办法活下来的，对，不可能，因为你的这些东西 ，OK， 都会被变成 acetyl-CoA。那变成 acetyl-CoA， 你没有办法回去 pyruvate。那你要知道，没办法做 pyruvate， 你没办法做 glycogen， 你没办法做很多 amino acids。OK， so as a result, it just will not work. Now, very quickly, these are the two reactions for glyoxylate shunt. You have, last slide, right? You have isocitrate lyase. Essentially, what it's going to do is it will cut this bond right here. Okay, it's going to cut that bond right there. And it's actually a very simple reaction. The reverse makes total sense because you can deprotonate this guy and then have it attack that guy, and then they form, right? So this guy, isocitrate lyase, converting isocitrate to succinate and glyoxylate, okay? And then succinate can go and become OA, okay? And then glyoxylate can then come in and react it with another acetyl-CoA, okay? And then you'll form this thing called malate, okay? So malate can, of course, malate can also go to OA, right? And malate synthase, again, it's a carbon-carbon bond formation, right? And it's a very useful thing. Okay, so this is the glyoxylate shunt. Right. So now, before we leave, real quick, what is the net reaction of TCA cycle? TCA cycle. 来 net reaction of TCA cycle. 所以让我 summarize 一下好不好？ So TCA cycle net reaction is what? 1 acetyl-CoA. Oh yeah, so you can have OA becomes 2CO2 and of course OA. Okay, so OA will cancel. And reducing powers, reducing cofactors. How many NADH? How many FADH2? 2 NADH, 1 NADPH, 1 FADH2. Right? I think right? 是一个 NADH 还是两个 NADH? I think it's 2 NADHs. Yeah, 2 NADH, 1 NADPH. Isocitrate dehydrogenase generate NADPH, whereas the alpha KG dehydrogenase produce NADH, and then malate dehydrogenase produce also NADH. Okay, but generally speaking, it depends on organism. Some organisms have a NADP-dependent malate dehydrogenase. They will actually produce NADPH. Okay, but that's just the details. Okay, so this is the net reaction of TCA cycle. Essentially, hold on, before we leave, real quick, how many electrons is this? 因为你看我这边有4个 molecule， 对不对？我有2个 NADH， 一个 NADPH， 然后一个 FADH2。那6是怎么弄来的？7个、8个、8个。8个 sounds right. Okay, why? NAD carries 2 electrons. Okay, NADH carries 2. So you have 1, 2, 3, 4. Right? NADPH also carries 2. 5, 6. FADH2 also 2. Okay, 7, 8. Okay, so it carries 8 electrons. Right? Why is it that it's not an odd number? Okay, 它为什么不会是奇数？因为如果是有奇数，那你就会看到一个 molecule 上面有一个圈圈，有没有？那个东西叫什么 ？radical。Okay, 如果有一个 electron 在那边飘的话，那个东西叫 radical。但是这边没看到嘛 ，right？ 没看到哦。Okay, so net reaction 一个 acetyl-CoA， 2个 CO2 出来8个 electrons。Okay， 那这个8个 electron 分别是2个 NADH， 一个 NADPH 跟一个 FADH2。好吗？Okay, so that's it.